development is concerned, if you really want students to develop a hypothesis, it does take time, and it's embedded usually over the first 3 or 4 sessions. So I'm going to show you the way I approach it. And you mentioned the course source paper that we published a few years ago, where we use bioinformatics and molecular visualization. There's another course source paper about to come out. Just sent the proofs back today, so it'll be out in the next week or two, with Betsy as the lead author. And Betsy developed a really nice little worksheet for hypothesis development that again builds off of over several labs. And then there's another course source paper that's just been submitted, which is basically what I'll talk through, and it's got some worksheets associated with it that works really nicely for hypothesis development. So let me share my screen and see if I can make my slideshow work. Oops, I've got to share my screen. Okay, can you see my screen now? Somebody give me a thumbs up.
Yep.
Okay, great. So I'm going to talk about it the way I do it, and the way that seems to work really well. In fact, actually, we're just doing the last part of it this afternoon with my lab this semester. And I like to think about it from the perspective of, okay, you know, we've got some sort of structure that we want to make a hypothesis about. And how do you go from structure to hypothesis? And then an important part of that is, how do you go from hypothesis to predictions, and then to the proposal for what experiments you're going to do. And we step through that as you'll see. And beginning. So this is based upon this sort of, you know, that you've heard me talk about before, I'm sure, the essential components of research that need to be in your cure. And if I can look at the right-hand side of the screen here, you know, we always start with a discussion of relevance. And I think it's really key to getting the students engaged, to let the students take ownership from a very early stage in this, and let them think about relevance and develop relevance, and then explore the scientific background building into the hypothesis development. Now, you know, you mentioned you were thinking about either post-translational modifications or inhibitor design. You know, you can set that up in the relevance section so that students are thinking the way you want them to think, but they're doing the thinking and they're building, you know, the background that's going to lead to the hypothesis development. So I always start this discussion after we've talked about relevance, because, you know, I explained that we're going to do this over a 2 or 3 lab period. And we talk about what a good hypothesis is. You know, a hypothesis isn't something you just look up in a dictionary and come up with a hypothesis off the top of your head. It's usually, you know, really good if you've actually discussed with the students what a good hypothesis is. And, you know, we talk a little bit about sort of how science approached, you know, hypothesis, and really the way, you know, you can never prove a hypothesis correct. You can prove a hypothesis wrong, and you can eliminate reasonable alternatives. And I want my students thinking about reasonable alternatives all the way through, because that's a critical part of doing good science. And, you know, have them understand that you can collect evidence that's consistent with a hypothesis, or it can prove it wrong. And then we talk about sort of, you know, based upon prior observations. It may be observations already in the literature, it may be prior observations in the lab or whatever, but there's some sort of building off of prior observations. Has to be original. If you already know the answer to the question, it's not really doing science. And then more importantly, it has to be testable. And that's why we have a strong emphasis of linking hypothesis to predictions. And that, of course, leads to it being falsifiable. So because, whatever the projects in the MCC are, at some point you're working with a protein. And we start from a very general discussion. And this works whether it's first year students that have not had any biochemistry or it's upper level biochem majors. They always tend to forget that proteins only have two things in common. Okay, they have structure and they bind something. I mean, there is not a protein on the surface of the Earth that doesn't have some form of structure. There is not a protein on the surface of the Earth that doesn't interact with something. And if you start with that, you can actually build a really good sort of infrastructure for a hypothesis. And so we go through a little bit of sort of basic stuff about protein structure. And then we talk about what the structure of a testable hypothesis is. And this is something I referred to in the past as the rule of threes approach to hypothesis development. And the first rule of three is that there are three somethings I want the students to think about. Something, some aspect of the protein or ligand structure does something. It interacts with something, which is some aspect of the, if it's maybe a mutant that is involved in substrate bind or residues involved in substrate bind or whatever. But get them into that thinking of, if you think about some aspect of structure and you think about what it interacts with, and then you think about what that interaction leads to, whether it's catalysis, affinity, specificity, whatever has come out of this sort of original relevance and big picture thinking, you've really got, the first stage in developing a hypothesis for any project. So the three somethings. And then we've got a worksheet where, again, it sort of gives the background, because repetition is always good with students at any stage. Right? It's actually always good with me too. If you tell it to me enough, I might actually understand it sooner or later. But, we build this from the relevance and get them to write, what their overarching big picture, what is the role of the active site loop, just as an example. Put in anything else you want in there. Then we come down to we narrow the question, what's the appropriate scientific background, and that usually ends up in this, active site loop. Well, what is the role of residue X in substrate specificity, say? And now they can do their three somethings, and the worksheet just has space for them to write down their three somethings. And at that point, they've got a hypothesis. Residue X interacts with whatever it is, and it does something. You've got a testable hypothesis. And then we move into the from hypothesis to predictions. And this is in the rule of threes motif. This is the three questions. And the three questions are, what do I need to measure? What will I change or perturb to test my hypothesis? And how will I know whether or not the change is significant and consistent or inconsistent with my hypothesis? And then the worksheet is, the overview just has some examples And then there's a worksheet where they have to, again, write down their 3 questions. And at this point, they've now got a hypothesis, they've got a way to test that hypothesis, and they can move on, to developing their proposal. Once they've identified, and the most important thing is to identify what they need to measure that really fits with their hypothesis. It might be binding, it might be KCAD, it might be the structure of the protein, it might be the interaction of the protein with another protein or whatever. But, they're focused now on what they need to measure. And that just keeps everything rolling in the right direction, and it's nice and simple. And with that, I'm going to stop and see if you have any questions.
So my first question is how does this spread over? So is this a worksheet you do in lab, in class? And you said that you kind of have it embedded in your course. What are you doing with the students to get them ready for this process?
So usually I start every class with usually maybe a 5-minute overview of where we're going. And then, if we're going into the hypothesis development, and I always try to make sure there's some actual wet lab work as well as dry lab work or no lab work, but computational work or whatever, in every class. So, in the early part of the semester where they're learning how to do a cluster and they're learning how to use PyMol and those sorts of things, because we're building into a proposal, I have these things called snippets. And a snippet is where they actually have to write some words, create an image, a cluster alignment that illustrates what it is they're thinking of and how it relates to whatever. And then, they're learning how to assay the enzyme or something, because, that's something they've got to know. So there are lab skills they've got to develop while we're building into the hypothesis and the proposal. Now, my course, my upper level biochem course, is a writing course. And so I structure it because they have writing assignments that they have to do. And we try to build all of these sort of various aspects into those writing assignments. If they don't have writing assignments, if it's, sometimes when I've taught this course, it's not been a writing course, but then we have presentation assignments. They have to present a cluster and what it means and what it does, because it's all building towards the end of the semester where they're going to give a presentation anyway, or in the writing course where they're going to give a presentation and turn in, a written paper. And we build into that peer review, for most presentations, most writing assignments. There's either in-class time peer review where they anonymously review one another, the assignments, and we discuss it. And we talk about how peer review is a critical part of the scientific process. I mean, I learn far more when somebody tells me I'm doing something wrong than I ever do when somebody says, oh, that's great. So peer review is important. I'd much prefer somebody criticize what I'm doing than tell me I'm doing it just wonderfully, because I learned nothing one way and I learned something the other way. So, we build that into the process. But, depending upon where we're at and what the stage and what the amount of lab work they've got to get done to get to the next stage of the project, some of these assignments are, post-lab assignments. And, we talk about it, we get on with the wet lab work or the whatever. And in my world, wet lab and computational work are the same thing. They're all experiments. And, we use a combination of them throughout the semester. But sometimes we've talked about it, now turn it in before the next class period. Does that help any?
That does. One of the things I'm struggling with is we have a strange setup where all of our courses are just one. There's no 3 courses, 4 credits. And so you have to be mindful for how much work students have to do for lab outside of lab, because they're getting as much credit as an English class. And so anything I ask them to do for a lab outside of class, I have to cut back on learning new course material, or and so I'm struggling with how to balance that, because it sounds. Oh, go ahead.
I was going to say, I'm very sympathetic to that perspective, because students have lives, and often they have jobs, or they have whatever, they got to earn enough money to go to college or whatever. And so I try to make most of the major assignments part of the allotted course time.
Okay.
And I think that's a way to help students buy into things.
Okay.
You know, just loading work up on them after class is not really very fair, and not really very effective for that matter either, I find.
And as you go through that worksheet, do you give them examples maybe that previous students have done, or do you find that then they clump onto and want to do that example as their own?
Oh no, I usually give them an example that's totally unrelated to anything they think they want to do.
Okay.
Just to show them the structure and show them the process.
Do you have like exemplary ones that have been filled out that you share with other faculty?
Yeah, that would be helpful.
I can. I'm happy to collaborate with anybody on. And the course source paper that we've just got coming out now, the latest course source paper, is actually builds into it collaboration as part of the hypothesis development. And so, you know, that's fine. I'm happy to share. What I don't want to do is put things on a website where students can just download it. Oh, here's my hypothesis. I don't mind sharing with faculty, but if we're paid to teach students, we shouldn't be just spoon feeding them the information. So, you know, if anybody wants examples, just email me and I'm happy to share.
Right. Joe, you want to talk a little bit about how you do hypothesis development with your classes?
Absolutely. So I like the 3 things. So I'm just learning about that now. What I'm going to show here is a couple of little things, Kristen, and one of them is really kind of like how I've used a lot of what the course source and other things have had in kind of a very specific example. Listening to what I just heard Ellis, and I'm waiting to see that new paper and some of these sheets. Make maybe a specific teacher's guide, right? That would be something you could look at and come up with that development. So, you know, you talked about being PTMs. We've got 6 mitochondrial mutants and phosphomimic mutants, and I think we're at least 8 or 9 cytosolic MDH phosphomimics, and we don't know a lot of what's going on there. So, but we also have a lot more that we could talk about collaboratively or target, right? But what we said at the workshop, what we said virtually is start simple, right? Grab a couple and, you know, this is what we know. And then as Ella said, this, you know, you might be able to see some activities, but then what don't we know, right? What are those 3 things? And the beauty of the protein modification thing is, does phosphorylation change the structure? Does it change the activity? Does it change the specific activity, the affinity, and maybe even protein interactions, right? I mean, obviously that happens. And so you kind of almost look at it from almost a physiology kind of question when you're looking at phosphorylation. But so what I'm going to share with you is one of these kind of directed things that's found on the website. Okay, so first off, just a reminder, under teaching MDH cures, we have a bunch of syllabi, and some of them are the ones that Ellis had talked about. So you can kind of see how in the past, at least, we've stretched this out. I'm going to talk about one that's the third example, a biochem lab here. Now, granted, this was something you did twice a week, 4 hours each. I would not do that where I worked for 16 years before that, where I only had them once a week for 3 hours, right? So you have to kind of decide how you're going to pare it down. For a lot of folks, this is a 2-member, but if you look at the kind of blocks, there's some basic skills, there's some bioinformatics and protein modeling. I put them in blocks. You can see how I kind of did a three-day thing here for all of those things, were how I developed and integrated my hypothesis, but it was coming in different places or designing experiments, doing presentations and things like that. So that was here under teaching, under example syllabi, right? So now, and there's some of the papers that and links to the OUR website and Ellis's website, where they talk about a lot of, at least maybe the older version. When Ellis gets those other papers up, they will add those in there too, right? But so the specific example I have is a Word format, so you can look at it all you want. See how ugly my background is. I think I need to stop share and reshare so you can see what I just brought up. But you can at least see where it came from, right? And so what this is a more narrowed down kind of a combination of what I give to the students, and it really parallels a lot of what Ellis talked about. And some notes there in yellow for instructors. So kind of like I'm interjecting and, oh, by the way, right? So this is really how I did one of my last chances to do a cure in a lab where we were looking at protein-protein interactions, right? Not the phosphorylation, but now that I'm seeing what the stuff Ellis is, I'd love to create one. Maybe you want to work with me, or Celeste, you and I, or something, work on a version of something like this that really grabs a lot of the stuff, or maybe all the stuff that Ellis just shared, and and say, well, here's some ideas, and keep it under the password side, so only we can see it, right? But then help the folks that are doing these, using these phosphomimics or making new phosphomimics. So anyway, go
Great
ahead.
idea. I was just gonna say, yeah.
So yeah, maybe we could make like a specific one for phospho, right? And then as people develop these different programs, we can have a teacher's version of what Ellis is talking about, that really nice general approach, right? And so I give you kind of the instructions I gave the students when I did my cure last time. You can't find that kind of stuff on the syllabus, right? So here's your draft for writing an introduction, which involves a hypothesis. I kind of put it together because they're going to have to read background. They're going to have to do some modeling, maybe even some computational analysis as they come up with their introduction, right? And because this is part of a writing course, drafting and getting feedback and stuff like this. So here, I have students should be given information on how to write scientifically. Things that just hints that you might want to share with students that I've done. You have your own way of doing it. You could put stuff in or ask for other materials if you're doing. So that's what these yellow bolded sections are. And Lisa, when you were editing this, you were asking questions. That's a great question. Let's put this into a yellow instructor notes, right? Because what I use for teaching on how to write, it might be very different than somebody that's more organically inclined or whatever. So you can see exactly what we're doing from the course source. What is a good hypothesis? Using this for the students, it's based on prior tests. You saw all that, right? So it's really just a more focused approach to it, right? More notes for the instructors. Now, this is specific for when we were looking at MDH CS interactions. And what I gave to the students to have them drive, to look at the literature, to do some pi-mole work, whether it's pi-mole or CND3, or maybe even three-dimensional stuff that Celeste does. These are just questions guiding them. Start in general, what do we know? What are interactions? So these are guiding questions that I think if I were to do this now and see what Ellis did, use this for instructor mode to kind of mold them together. So you can think as an instructor, what do I want to tell my students to think about while I'm doing this in terms of protein interactions, or now maybe phosphorylation, right? And then that's where models in bioinformatics are you doing PyMol? Are you CND3? Are you doing, you know, other things? You're using HawkDoc, using, you know, other things. You don't necessarily have to dock things. We did for the interactions. And so these were, again, prompts for the students to study the materials we had, the papers, to use some of these activities to start increasing their understanding of what interactions are, what are, you know, what are the interfaces. So you can't, to help them develop the hypothesis, they got to get some more background. And I could see if we do this for a phosphorylation thing, we could do the same kind of thing. What is a kinase? You know, depending upon where they're at in their biochemistry, kinases might just be something they heard in a first year biology class or something, right? And so all these could be then merged into a very specific thing for protein phosphorylation. And then what I like to say is, what is the gap, right? We all write our grants in different times. What is that gap in knowledge, and how it is my question developing a hypothesis to fill that gap? And so these are, again, are prompts that I give the students that are very much oriented towards the project, but they're also general enough it can be adopted to anything. And I'm, you know, again, I really am looking forward to trying to find some time to remold this into what we saw that Ellis has been putting together with Betsy and some others. Tell them to draft their hypothesis, start, and this is my mode, but I would now definitely include the three things, right? But start with, because of this, see number 1 or 2 above, we don't know this, the gap. Therefore, X is important and will cause or be important for these things. Some hypotheses are, and then I just walk them through the, so again, you know, this is more what I give the students. And what Ellis had was how you would want to then do this. So this is kind of a gap filler, if you will. And then I get to the same kind of thing. How do you then, if this is your hypothesis, it's got to be something that's testable. I give them ideas, right? They very general. So they can't say, oh, I'm going to go test the interaction. Oh yeah, how, what, which ones, what mutations? So this is not something they can say they're done with, but at least gives them a direction. Some faculty don't want to give them any direction. Well, that's up to you. You might say, I want to focus on this, right? So now you develop your question beyond that. And again, these kind of things as we develop them would easily be behind a password thing. And then I give you the handouts that I would use in that semester, presentations, peer evaluations, assessment, what I give them from what I expect them to do. And if you find this helpful, you can grab it. It's in a Word file. You can copy, adjust it. Nothing is there that I don't give the students ahead of time, right? And none of it is, so if they just copy paste what I gave them, they'll get an A. There's no way. I give some very explicit instructions on data presentations towards the end. I just feel the more I tell them, the less generic Excel background automatic results I get. Conclusions, number of slides, grading how I emphasize this. I don't have a complete rubrics for this. Mine wasn't that hardcore well enough to share, but from this you could develop your own hypothesis or rubrics for the entire semester. There. But I really think that using the stuff that we're seeing coming out now, it would be cool to create a protein phosphorylation, because we'll have 10, 12 different mutants, and there's at least 20, 30 others that are open, right? And so having this to share, to hand to other instructors as they're doing this, makes life simpler, right? At the same time, you see all the expansion that can go on.
Yeah, I totally agree. I think a worksheet, it helps focus the students, and they can follow along and fill it out as they go through. I think I totally love the idea of adopting Ellis's structure to the PTMs.
Yep. And again, putting that aside, so in a place where the students can't see it, because maybe you don't want, so for instance, Lisa does